Okay guys, your final video for your exams. Here we go, multiple choice topics. This will be done on either Monday or Thursday, depending on which day you come on. And I'm going to go pretty fast, so hopefully you pay attention. You're going to be tested on the scientific method. And you need to know that the steps of the scientific method, you of course will observe, then you'll form a question, and then you are going to make your hypothesis, you'll perform your experiment, you'll analyze your results, and you'll write your conclusion. Your hypothesis, of course, is an explanation of what is tentative and it needs more investigation, whereas a theory no longer needs investigation and because it's supported by a bunch of evidence. And you need to know the difference between the independent variable and the dependent variable. The independent variable is what you change, the dependent variable is what you measure your results in. You'll have questions about properties of water. Water is an excellent solvent. It does great heat retention, meaning it will hold heat and it will heat up quickly. It can be an acid or a base or be neutral. And a lot of these questions that I'm talking about right now came from old tests. So if you go on Blackboard and read over those questions, that would actually be an excellent way to study. pH, if you are a basic solution, that means you have more hydroxide ions and you have a pH of greater than 7. If you're an acidic solution, you have more hydrogen ions and you have a pH of less than 7. There are different types of bonds. There's a nonpolar covalent bond, a polar covalent bond. Nonpolar covalent means it shares electrons but does not have a charge. A polar covalent bond means that it shares electrons but does have a partial charge, like water. Partially positive on one end and negative on the other. Your ionic bond is a bond where you are transferring electrons from one element to the next, and a hydrogen bond is where you are attracted to each other because of partial charges. So a partial positive is attracted to a partial negative, kind of like a battery. Biological molecules, the only molecule that he really had on the test was a question about a nucleotide, which was, I think, on one of your most recent tests. A nucleotide is made of a sugar, a phosphate group, and a nitrogenous containing base, or a nitrogen base. Characteristics about viruses. We talked about some, but we didn't go into a whole lot of detail. But viruses are not alive. They are not made of cells, and another fact that you need to know about viruses is they only reproduce when they are inside a living host cell. So your question will be like, all the following are factors of viruses except, and you'll have to pick. And then there's another one about the reproduction one. I think there's 2 or 3 questions about viruses. Organelles and their function. These are in different questions. Some of them are like multiple choices in the same question, but if you know these facts, then you'll be able to answer the question correctly. Nucleus, of course, is a control center of the cell. The chloroplast does photosynthesis. Mitochondria does cellular respiration. The Golgi body is responsible for modifying and packaging proteins and lipids, because you've got a question about proteins too, so you need to pay attention to that. Oops. We've got, where was I at? Golgi body ribosome synthesizes proteins, ER modifies proteins. And another question about organelles and types of cells is eukaryotes have a nucleus surrounded by a membrane, but bacteria does not. That's its own question. You need to know the 3 parts of the cell theory and what is not part of a cell theory. That was one of your questions from a previous test. Cells are the basic units of life. All living things are made of cells, and cells come from pre-existing cells. You have questions about cellular respiration. In cellular respiration, most of the aerobic respiration, most of the ATP, that's what it should say, most ATP in aerobic respiration results from the electron transport chain. You get very little of it in the other steps. The electron transport chain plus the other steps makes approximately 36 to 38 ATP. The role of the oxygen in this whole process of aerobic respiration is to accept those electrons as they go through the electron transport chain, and that is, those are released by oxidizing glucose, and we make water as a result when hydrogen joins with the oxygen at the end. We also can do fermentation without oxygen, and animals in fermentation will make lactic acid and plants make ethanol, and that is because we metabolize pyruvate and it doesn't go any farther. In the reverse, the flip side of that is photosynthesis. Photosynthesis will use light energy from sunlight, and it will convert carbon dioxide into organic compounds by splitting water. Those light reactions will make things like, will make the products of ATP and NADPH. Remember, that's part 1 of photosynthesis. Then that ATP and NADPH goes into part 2, which is called the Calvin cycle, and it will use Rubisco, which is an enzyme that the plant cell makes, and it will take in carbon dioxide through the stomata and join it with RUBP, and it will do that to synthesize simple sugars. Again, these are all different questions on your test. I just grouped them all together here. Oxygen is produced as a byproduct of splitting water. And the oxygen produced here from the light reaction step will actually end up as water again in cellular respiration, because the oxygen becomes that last electron acceptor and it will join with hydrogen to make the water. So oxygen comes from water and it goes back to water. It's a full cycle. Cellular energy is the quick form of it is ATP, adenosine triphosphate. And enzymes help to lower activation energy in a cell. They do that by having an active site where substrates are oriented to fit together and so they can react together quickly and easily. You need to be able to read an enzyme graph. I'll have to put one up later, but you're going to have a graph and be like, this is what the energy would be without an enzyme. Now, which one of these other lines, there's one like this and there's a couple down here, which one of these other lines would happen if you had an enzyme present? And of course, it's the one that comes up and then kind of levels off at its reaction rate. So that's the one that you want to find, the one that comes up but it's lower and then it levels off. Specificity, it's a hard word for me to say, due to the shape of the enzyme and the substrate. So it's like a lock and key. Remember, enzymes only work in one specific reaction because of their shape. You have questions about membrane. One question about membrane transport, and it's like, which type of molecule is least likely to cross the membrane? And it's got different molecules. The fact that you need to be able to pull from to answer that question is phospholipids will not allow large molecules to cross, like really big ones, or charged molecules. So a molecule that has a charge will not go across that membrane either. There are questions about the cell cycle. And again, all of this is review. Make sure you know that, there we go. Make sure you know that interphase is the part where all the other stuff that takes place when it's not reacting. In interphase, it's also where DNA replication will take place. You need to know the steps of mitosis, so remember PMAT. And you need to know specifically in the prophase part of PMAT is where the condensation of chromosomes occurs. The nuclear envelope will disappear and the spindle fibers will begin to attach to those sister chromatids. Another fact you need to know is daughter cells will have the same number of chromosomes and DNA at the end of mitosis as what they did at the G1 step of interphase. And you need to know that chromatids are replicated chromosomes attached together at a centromere. I'm trying to get through these all in one thing, but I think I'm going to have to take a stop here. So my video will upload, and when we come back, we'll talk about genetics.